Hello friends, now last lecture we have discussing about the new chapter biotechnology. In this chapter, this biotechnology, it is most important branch of biology. It is one of the applied branch of biology, and it is a study of several application of this technology. There also include the microbiology and other branches like the chemical biotechnology or some genetic microbiology. They are also including in this biotechnology. And this biotechnology, it is most important for using some microorganisms, and this with the help of these microorganisms produces some products, or these products they are also useful to human welfares. And this biotechnology, it is also the two types of biotechnology. Means first one the oldest biotechnology, and another is the new biotechnology or modern biotechnology. Oldest means traditional biotechnology. This traditionally biotechnology, the various uses for this traditional use of this biotechnology means one of the basic process like the fermentation, and these fermentation techniques using some microorganisms, and they can prepare the curd or various food products are produced in the traditional biotechnology. Ghees, vinegars, these are the some traditional biotechnological uses. Now some modern biotechnology. In this modern or new biotechnology, it is also involves the most important term like the RDNA technology, recombination DNA technology. Means this manipulation of the gene, insert the new gene from organisms, they are including this RDNA technology. And there are some major features of this biotechnology. And now this some principle we are discussing, some principle of biotechnology or processes. Means one of the genetic engineering and another one is the chemical engineering. The genetic engineering is also useful for the alternation of the gene or the alternation of genetic material. And this one of the manipulation of the genetic material or directly into the cell. This is a genetic engineering. And another one, the chemical engineering. This chemical engineering maintaining the sterile environment for the manufacturing the variety of useful products. Now these some useful products of chemical engineering like the antibiotics, vitamins, vaccines, or some theoretical uses of this biotechnology. Now the next concept we are discussing in this video, some tools and techniques for gene cloning or RDNA technology. There are some basic tools are using for this biotechnology, and these tools like some RDNA technology or some different instruments are using for this biotechnology. Means this RDNA technology or gene cloning or RDNA technology means the manipulating the gene, or the gene are insert from the another cells, new gene are synthesizing. And now these techniques are also used for the different instruments. These techniques are involving some different instruments or devices are uses for this technology. Now these some macromolecules are using for this technology. Means this some instruments of the macromolecules like DNA, RNA and proteins. These are the macromolecules is very important tools for the genetic engineering. Means DNA is a genetic material, deoxyribonucleic acid, RNA and proteins. These some macromolecules are also important tools for the genetic engineering or gene cloning or RDNA technology. And another one, the electrophoresis. This is one of the important for the separation of charged molecule. They are applying of the electric fields. This electrophoresis means the separation of the charged molecules, and they can applying the electric fields this electrophoresis, it is also separate the small segment of DNA fragments, and using these techniques or using this electrophoresis, the faster process, and that gets separate the gene or separate the DNA molecules. And also used for the agarogel electrophoresis or PAGE or also SDA, PAGE. These are a different method of the electrophoresis are using for the tools of or techniques of genetic engineering or gene cloning process. That the different tools are using for these instruments are using for the RDNA technology. Now the next concept is a PCR. These new machines like the PCR is most important tools or gene cloning process. The PCR technique. PCR. This polymerase chain reaction. Polymerase chain reaction. This PCR techniques or this PCR it can separate or the generation of billions of copies for desired DNA. They can generate the billions of copies. Billions of copies are generated. The DNA molecules are generated. Billions of copies are generated with the help of this machine or these polymerase chain reactions. And these terms or they are also used for gene cloning or RDNA technology. Gene or multiplication means gene multiplication takes place in this machine, PCR machine. It takes place in vitro condition. In vitro condition means the aseptic condition, and this process takes place, and it is this PCR technique is studied in this 1985. This machine is discovered or they can made important discovery of this machine in 1985. Carey Mullis, this scientist discovered this machine, and later 1993 this Carey Mullis was awarded the Nobel Prize. And this PCR machine they can generate or this machine is useful for the generation of billions of DNA molecules or production of the DNA molecules takes place through this machine. This process of PCR is completely atomoded. Automatically these DNA copies are produced. Automated machine, and they are involved the thermal cycles and for the denaturation and regeneration of the double stranded of the DNA. This PCR machine or this PCR technique device, it is also called the thermal cycle. This is also called thermal cycle. This PCR machine, and this device, there are very important requirements are required for these PCR techniques. The thermal cycle or this PCR is a in vitro techniques. They are applying the several requirements. The first one requirements of this machine are utilizing the one of the DNA molecules are most important requirement for PCR techniques. Some requirements. The first one, segment of DNA molecule. First important requirement is a DNA, and it is a short segment of DNA is important for these techniques, and they can be multiply these DNA fragments. And also important another requirement, 4 types of nucleotides. 4 types of nucleotides. This another requirement. Nucleotides are very important. These nucleotides that is nucleotides, that is ATP, GTP, TTP and CTP. Means these are the 4 types of the nucleotides deoxyadenosine triphosphate, guanosine triphosphate, thymine triphosphate and cytosine triphosphate. These are the 4 types of the nucleotides are also required for this process, and this collectively all these nucleotides, it is called NTPs. It is called NTPs. This NTPs means it is a deoxyribonucleosides triphosphate molecules. These triphosphate molecules, they are also required for this process. The another one requirements, tag polymerase. This enzyme is most important. Tag polymerase, or also the Mg++ ions is also required for this process, and 2 primers is also important requirement for this PCR techniques. Now let's the mechanism of PCR technique. These are the requirement are very important for these machines or PCR techniques. And now the mechanism of PCR. Mechanism of PCR machine. There are involves the 3 steps. The first step is a heat denaturation. Heat denaturation. In this mechanism, second step, annealing. And this third step, this heat denaturation, annealing and polymerization, or this third step, polymerization. These 3 steps are involved in this mechanism. This PCR techniques, it is the most important, and this machine they are started the PCR with the help of this requirement like DNA segments, tag polymerase, some primers. All these requirements, all these molecules, they are mixed with these machines, and later this machine is operated and perform the some steps. The first step is a heat denaturation. In this figure, let's study these 3 steps. The first one copies of desired gene, and this first step is a denaturation. These are the 2 strands of the DNA, and these 2 strands of DNA, there are some complementary bases are present, nitrogen bases are attached to each other, and these strands, complementary strands is found, and this first step mixture or mix this material, all these requirements, and later this machine, the heat denaturation process is started. And this heating process, denaturation process, is about 90 to 98 degrees centigrade temperature. This first step is required. At that time, they get separate the 2 strands of the DNA molecules. For the desired DNA, means this double stranded of the DNA, there are present some hydrogen bonds, and heating process is takes place 90 to 98 degrees centigrade. Heating is takes place. That time this hydrogen bond is break down, and this DNA molecule they get separated. Means this separation of these 2 strands, they get separated to each other. And now next step is annealing. The second step, annealing of primers. Now this separated stand, these two stands are separated, and the primer is very important. The mixing is allowed to cool this machine, or this temperature is low, like 40 to 60 degrees centigrade in second step. That step, they are comparatively, this permits the primers, and this pairing of this primaries takes place to be the complementary sequence of the DNA stand, and this step is called the annealing. When this complementary stand of the DNA, they can separated heat processing, and later these two stands are separated. In the annealing process, the stands get separated to each other, and later in this step, the replicating DNA stands using the annealing is a primer. This primer is most important. They are attached on this stand, the 5' or 3', and in this stand, this primer get attached. This primer is attached for the second step, annealing step, and in this step, the pairing of primer is occurring in the second step. Now the third step, last step of this PCR mechanism or polymerase chain mechanism, the third step, polymerization. Now this polymerization step, in this step, the replication of DNA stands, and this temperature about the 75 degrees centigrade, 70 to 75 degrees centigrade temperature, and allow this thermostable enzymes are used for this step, the Taq polymerase. These enzymes are using this Taq polymerase, the replication of DNA stand, that time using the Taq polymerase enzyme. This Taq polymerase enzyme is the most important. They can using the single stranded DNA as a template stand, and some adds of the nucleotides. Addition of the nucleotides is takes place. These two primers are formed, and this primer later the addition of nucleotides is takes place in these two strands. These two strands they get separated, and replication of DNA, that using the some Taq polymerase enzyme, that time this process nitrogen bases or they can appear in these stands. And now this primer, the last step is a polymerization extension, and this extension it is takes place between the 2 minutes, and this step duration is a very low, and this duration Taq polymerase, this enzyme is most important, and this Taq polymerase enzyme, it is a thermostable enzymes, and they can using the bacterium. This enzyme is collecting some bacteria like Thermus aquaticus. This bacteria, they can forming this enzyme Taq polymerase. In this thermostable enzyme is using for this step, the polymerization step, and this polymerization it is the last step of the PCR techniques, and that these two copies of desired gene means renaturation is takes place. These two copies of the desired DNA molecules is formed in a short periods, and the cycle is continuous to produce the double copies of DNA. And this revision of these cycles or revision of these three steps, heat denaturation, annealing and polymerization, 3 steps revise, means after these 3 to 4 times this cycle is takes around, and that times they can begin the second cycle. DNA again and again these DNA molecules are double structure form, and they can duplicate, multiply these DNA molecules within a short periods, and it is the most important mechanism of this PCR. Means this PCR means it is one of the important instrument are utilizing the duplication of DNA, means the regeneration of the DNA molecules in this machine, and this PCR is the most important machine for the multiplication of the DNA molecules, means the formation of DNA molecules within a short period this machine is utilizing, and billions of copies are produced through this machine. These are the three steps of the PCR machines. For mechanism of this PCR, there are three steps. One is a heat denaturation, second annealing, and third step is a polymerization.